Hello all, this is me Rajneesh Khambhati and this is my channel BioQuest Academy. I'm taking online classes also, okay, and this is my name. Now we are going to solve today this paper of biology part A MCQ. This is paper set number 1. Okay, this is the paper from March 2026 biology paper. So let's begin. Okay, now see, this is question number 1. Let me zoom it for you. Now this is question. Okay, this is not good over here, but I will tell you. See the question first of all. At the micropylar end, egg apparatus is constituted by the fusion of which cell? Okay, very carefully understand question. See, at the micropylar end, okay, where at the micropylar end, this is the position. The egg apparatus is constituted by the fusion of which cell? Means which cells are fusing at this end. Now, 4 options are given. Option A, B, C and D. 2 male gamete and 1 egg cell, 1 male gamete and 2 egg cell, 2 synergid cells and 1 egg cell, or 2 synergy cells and 1 male gamete. This is male gamete written. Now, before directly I will tell you from this diagram. See, this is a diagram of for this question. And let me tell you one thing, that in biology you require to learn the diagram very carefully. Okay, because from the diagram we can answer it. Now see very carefully. What do you see? This is, okay, a diagram where, okay, this is egg cell. This is egg cell, and this end is micropylar end. So at the micropylar end, the egg cell, okay, it is fusing with other 2 cells, which is that 2 cell. So you can directly see in the diagram 2 synergid cells. Okay, so 1 egg cell, 2 synergid cells, they will fuse together like this in the diagram. You see, where at the micropylar end. So from the diagram you can actually answer it. So from here, what can be the answer? Okay, 2 synergid cells and 1 male gamete. But over here, this is not male gamete, it is an egg cell, female gamete. So this is not the answer. 2 synergid cells and 1 egg cell. So this is correct answer C. Question number 2. What is the number of chromosome in primary oocytes and the first polar body? You have to give the numbers of chromosome where at the primary oocyte and the first polar body. Again, we can answer directly from this diagram. Let me show you the diagram. So this is what oogonia. Now, oogonia first undergoes mitosis, differentiation, and produces primary oocyte. Okay. Now, primary oocyte undergoes the first meiotic division and gives you secondary oocyte and the first polar body. Okay. Now, see here, the primary oocyte is having 46 number of chromosome, and the secondary oocyte along with the first polar body, they have 23 number of chromosome. Why? Because they undergoes the meiotic cell division, and we know that during the meiosis, okay, the chromosome number will become half. So from the diagram you can see, we can answer it here. See again, what do we require? Number of chromosome in primary oocyte and first polar body. So primary oocyte is having 46 number of chromosome and the first polar body is having 23. So 46 and 23. Answer is B. Diagrams are very important for you. So I would suggest you that everybody should go for the diagram first. See, normally students are, they are reading theory, they are practicing the theory. That is good. You require that. Okay, you will be doing some practice of MCQs and other things. But diagram based questions are also very important. Okay, so go for the diagram. Learn diagram by heart, everything, and properly. Okay, let's move to the next question. Question number 4. Read the question. Read the question. What do you see? Which of the following is an example of hormone releasing IUDs? You need to answer hormone releasing IUDs. 4 options are here. Multilode 375, Lipslubes, LNG20, progestogen. 4 options, you have to choose one. Now, what is the answer? Answer is LNG20. This is your answer, LNG20 from the textbook. Okay, from there you can understand. Say, hormone releasing IUDs are progestasert and LNG20. Okay, this is the answer. They are hormone releasing IUDs. Now, why I am doing this? Okay, the cutting of textbook, because you can understand, so that you understand that the line by line NCERT is very important, very must. Okay, question is question number 4. Which disorder is caused by autosomal dominant trait? Now, here 4 options are given: myotonic dystrophy, sickle cell anemia, phenylketonuria, hemophilia. But before we answer, you should know everything. Okay, if you know all the options, okay, so that it will be easy for you to answer. Sometimes if you know 1 or 2 diseases, okay, what you can do is you can do 50-50 when you know half answer. Okay, that this 2 cannot be the answer and this 2 can be. So in that case, this will be helpful. But over here, to just explain this, I have written everything. Okay, in the short form of explanation. See, myotonic dystrophy is what? It is autosomal dominant. Now, what we require? Autosomal dominant. So, myotonic dystrophy is autosomal dominant, but that means this is our answer. Answer is A. Here, then what about sickle cell anemia, phenylketonuria, and hemophilia? So, here, sickle cell and the PKU, they are autosomal, but they are recessive. Hemophilia is what? It is a sex-linked recessive. Then phenylketonuria, again it's a inborn error of metabolism. Okay, where the thalassemia, okay, but it is not in here as option. But thalassemia is what? This is also an autosomal linked and that too recessive. Okay, this is the question from chapter 4, principles of inheritance. Okay, but answer is myotonic dystrophy. What it is? It is an autosomal dominant trait. Next question is very easy. Question number 5. Read the question. If in any double helix of the DNA, the percentage of adenine is 35. What is given? A is 35. But here percentage. Then what will be the percentage of cytosine in that DNA? Now the question is very easy, but before that you should know the basic behind the DNA. Double helix. Here see, DNA, but DNA is double helical. It is very clear cut given. So when A is 35, now we know A is always equal to T according to the Chargaff rule. Okay, means if adenine is 35, obviously T is also 35 because adenine will bound with the thymine with double bond, double hydrogen bond. So adenine is equal to thymine is 35. Now total this will give me 70 percentage. Now just simple, out of 100 minus 70 percentage, what will be the answer? 30 percentage. So 30 percentage is remaining. Now A and T we know. What is remaining? Guanine is binding with triple bond with cytosine. So G triple bond C. Okay, so 30%, that means make it half, 15, 15. That's it. So the remaining G and C ratio will be 30 percentage, but if I divide it, this will become 15 percentage. Okay. Okay, then let's move to the next question. Now again, this is the question you can answer from the diagram. See, diagrams are very important. Here, which gas is not used in, very carefully read, not used in Miller's experiment? Options are H2S, NH3, H2O, CH4. Directly go for the diagram. This is Miller's experiment and he used CH4, NH3, H2O, H2. He used it. But the question says not used. Simply, okay, NH3 used, H2O used, CH4 used. That means answer is H2S. Getting the point? Diagrams are very essential. Next question. Question number 7. Fill in the blank is very effective, sedative and very useful in the patients who have undergone the surgery. Options are heroin, marijuana, barbiturates and morphines. Here, this is not the answer. Okay, it's my mistake here. See, this is again cutting off the textbook. Mental illness like depressions and insomnia are often abused. Morphine is very effective, sedative and painkiller, and it is very useful in the patients who have undergone the surgery. That means answer is morphine. Where is the morphine? D. Option D is your answer. This is mistake. Okay, don't see that. But again, this is from textbook. Now again, line by line textbook. Okay, important. And that is why every teachers are saying that NCERT, you have to go through the NCERT line by line. It is very important for you. Next question. Question number 8. Here, match the column A with column B and choose the correct option. Column A, column B, let's just see. Lactobacillus, Trichoderma, Polysporum, Streptococcus and Saccharomyces cerevisiae. This is options. And over here, A, B, C and D, cyclosporin A, clotbuster, ethanol and curd. Now, once you read, okay, once you have read this, then just try to connect two ends, whatever you know. Okay, sometimes it happens that you don't know everything. But for example, Saccharomyces cerevisiae, okay, we all know that we use this to make this. That's it. And we also know that Lactobacillus, okay, we use it to make the curd. Okay, two things we know, for example, very easy. Now other two, if you know anyone, okay, you can do that. But now try to, let's do what 50-50, okay, by chance. That if you don't know the answer, then how to do? When you know only two, then what to do? Here see, 1D, 4C. So 1D, 1D and 4C, 4C, 4C. Do you see? 1D given into B and C, whereas 4C is given into A and B. Means over there, this is only option. This is only option where it is matching perfectly. So directly, without any thinking, this is your answer. But proper answer here given that we use lactobacillus to make the curd. We use trichoderma to form cyclosporin A. Now, where do you see cyclosporin A? Okay, this is cyclosporin A given by this is here. So 2 with A. Now you see 2A and 3B. 3B. So everything matches perfectly, but you have to understand every time your luck is not going to work like this. Okay, but you can try this if you don't know or if you half answer in certain cases. Next, here, which of the following restriction endonuclease enzyme is discovered first in biotechnology? Means first discovered RE. Answer is Hind2. Again, this is from line by line textbook, but here the first restriction endonuclease is Hind2. See, line by line questions. Okay, so go through NCERT, and let me tell you, I am going to put NCERT line by line explanatory video for you on my channel. So please do subscribe, do share, and do comment, please, if you like this video. Okay, now next question. I hope that I am properly audible and you are able to see this. Question number 10. At present, how many recombinant therapeutics have been approved for the human use the world over? Now, means in worldwide, how many are means recombinant therapeutics have been approved for the human use? Answer is 30, and it is coming from here. See, at present, about 30 recombinant therapeutics have been approved for human use. Okay, human use the world over. Kind of different sentence, but this is the okay as it is. See, as it is, no change at all, no change. So and in India, 12 of these are presently being marketed. Now, who knows, next year maybe they can ask this question. Okay, how many recombinant therapeutic drugs are being marketed presently in India? So this is your answer, 30. Remember, 30 therapeutics have been approved all over the world, and 12 out of that 30 are presently being marketed in India. Next question here. Question number 11. Which type of population interaction occurs between sea anemone and the clown fish? Now this is again very easy question, and you know that sea anemones are something like this. Okay, they have tentacles on them. They have tentacles. Okay, they have tentacles. These tentacles of the sea anemone, okay, they have poison in them, like this. Okay, they have poison glands and they have poison sting. That is how they catch their prey. So normally they are very dangerous and poisonous. But in this sea anemone, the clownfish is residing. Okay, the clownfish and the sea anemone, they have close association. The clownfish is living inside. Okay, they live The clownfish is living not with but inside this sea animal. Now this is the kind of relationship. Okay, that means interaction. Now this interaction is what? Mutualism, parasitism, commensalism, or amensalism. Answer is over here. See, the sea animal that has a stinging tentacles and the clownfish that lives among them. The fish get protection. Okay, this fish will get the protection from other predators which stay away from the stinging tentacles of the sea animal. A sea animal does not appear to deliver any benefit by the hosting clown fish. That means in this relationship, the fish is getting the benefit. Means I am writing plus, but the sea animal is getting nothing, like zero. Okay, it is benefit nor harm. That means remaining neutral. Now this kind of relationship is known as commensalism. Okay, so answer is commensalism because clownfish get the protection, sea animal remains unaffected. This is again NCERT concept. Okay, from the book you can see here. Another example of commensalism. Answer is here. Next question is question number 12. Which of the following is not related to the structure of the ecosystem? 4 options: productivity, degradation, conservation, energy loss. What can be the answer? Not related to the structure of ecosystem. Now, simple explanation. This is from the chapter 12, ecosystem. The structure of ecosystem, okay, contains productivity, energy flow, decomposition, etc. Okay, these are in the structure of ecosystem, but the conservation is not there. Okay, conservation is not there. So conservation is your answer. That means, see, not that this is your answer. But energy loss, degradation, productivity, it comes in here. That's it. Very simple, very easy. Okay. Our next question, question number 13. As we move away from the equator towards the poles, fill in the blank species of the bird find in Colombia, species of birds find in the New York, and species of the bird find in Greenland. Now everything again, this is the cutting of the textbook provided. New York 105 species, Greenland with 56 species, and this is 1400 species of the bird near equator. So the answer is here 1400, 105 and 56. Oh, next question, question 14. Is it an example? Okay, so sorry. It is an example of persistent nucellus called perisperm. Means we can find perisperm in. Give example of it. So 4 seeds are given: castor, maize, beet, and the beans. Direct answer is beet. But from here you can see, okay, showing some seeds such as black pepper and the beet. The remnant of nucellus are also persistent. Means we can see the nucellus, okay, in those seeds. This residual persistent nucellus is called perisperm. So we can see perisperm into black pepper and beet. So answer is C, beet. Next question 15. The first movement of the fetus. See, the first movement of the fetus occurs in which month? Means during the period of pregnancy. Okay, at what time exactly you can see the first movement of the fetus? So again, this is cutting of the textbook. The head are usually observed during the 5th month. By the end of about 24 weeks, there is a little one mistake by me, that is the line is above here, okay, in this cutting. By mistake I just cropped it. So, but answer is 5th month. Okay, answer is 5th month. During the 5th month, you can see the first movement of the fetus. That's it. Next, question 16. Which of the following is not correct? Question number 16. Which of the following is not correct for the contraceptive pills, Saheli? Now, before we go over here, see 4 options. Non-steroidal, take once in a week, low contraceptive value, few side effect. You can see here. Saheli is new oral contraceptive. It contains a non-steroidal preparations. It is a once a week pill with a very few side effect and have a high contraceptive value. So see, high contraceptive value, and we have to answer not correct. So this is option C, written low contraceptive value, but actually it is high contraceptive value pill. So answer is C, low contraceptive value. Now please mark this one thing over here again, and that is, in normal questions they are asking not. Some students are, without reading complete answer, they will try to answer it, but which is correct. But you see very carefully that in many questions of this paper they are asking this not. Okay, so very carefully read your question first. Next question 17. Who proposed the chromosomal theory of inheritance? Direct answer, you can see Walter Sutton and Theodore Boveri. Again, the answer is from here. Okay, textbook, directly line by line. But you need to read it full. See, Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of a factor they carried. And Sutton united the knowledge of chromosomal segregation with Mendelian principles, and then he called it chromosomal theory of inheritance. So who gave the theory? Walter Sutton and Theodore Boveri. Now next, okay, question number 18. Which scientist proposed the thought of central dogma? Who gave this? Francis Crick. Chapter 5, molecular basis of inheritance. This is central dogma. DNA will form RNA. RNA forms protein. Okay, and DNA also forms DNA, which is called as replication. DNA forms RNA, that is called transcription, and RNA forms protein, that is called translation. This is called central dogma, given by Francis Crick. Next, okay, next question, question number 19. Select the brain capacity of Homo erectus, Neanderthal man, and Homo habilis. There are 4 different options are given, okay, and the answer is B, 900 cc, 1400 cc, 650 cc from this cutting of the textbook. What do you see? See, Homo habilis with 650 So Homo habilis with 650. Then the Homo erectus with 900 cc. So Homo erectus with 900 cc of brain capacity. And in the end, here, the Neanderthal man with the brain capacity of 1400 cc. Okay, so answer is this. And D. 900 cc, 1400 cc, 650 cc. Next question, question number 20. Now if you see the question number 20, which of the following is primary lymphoid organ? Option A, spleen, thymus, tonsils, small intestines. From here, cutting of the textbook, lymphoid organs means these are the organs where origin and or maturations and proliferations of lymphocyte occurs. What is the lymphoid organ? It is a place where maturation and proliferation of the primary immune cells are happening. The primary lymphoid organs are, there are two organs, primary lymphoid organs, bone marrow and the thymus. Now, what is the question? Primary lymphoid organ. Okay, we have two, bone marrow and the thymus. Spleen, thymus, tonsils and the small intestine. Answer is here, B, thymus. Question 21, this is for general students. See the diagram and nomenclature it. This is again proper diagram based question. What is this portion? Head. This portion is collar. This portion is tail. This portion is plate. And this portion is pins. And these are prongs. Simply, okay, you just need to write. A is head. This is collar. C is tail. And this portion means pins. So head, collar, tail and pins. Answer is A, that is good. But what I am saying is, what if you know only few things? Okay, what if you know only few things, like very clear thing. A is head, clear cut. Okay, so you see where you find head, three places where you can find head. If you know any one of this, okay, so let's take about a pin. Means where do you see the pins? We can find pins at two places only, at A and at D. Okay. Now what if you know any one of this? Maybe collar or the tail. Okay. So means B is collar. Now where do you see the collar in the B? No. C for the tail. Where do you see? So you find tail at option A and option B. Okay. Head, tail and the pins. So maximum number matches into A. Okay, but this is question number 21A for the general students. Next question for the blind student, 21B. The two columns are given: leg, sap of palm, Propionibacterium shermani and the lipase. In the column B, laundry, Swiss cheese, toddy and increasing B12. Directly, okay, this, see, I just made it. Leg will give you B12, sap of palm is toddy, Propionibacterium shermani we use into Swiss cheese making, and the lipase we use into laundry. Simple. So your answer is A. Okay, here. Let's do it. LAB into B12. So 1 with D. 1 with D. Then Propionibacterium shermani into the Swiss cheese means B. And sap of palm is stored means C. Surabay A. Laundry. Okay, D, C, B, A. D, C, B, A. So answer is A. 21 B. This is for blind student only. Next. 22 number of question. If we cut the vector PBR 322, this is diagram of PBR 322 from the place of BAM H1. Now, where do you see the BAM H1? Here. Okay. Now, what they say, we have to cut the PBR 322 with the BAM H1 means at here and make rDNA from it. Then, for which antibiotic it remains resistant. Okay. You see, BAM H1 is inside tetracycline resistant gene, and PST1, PVU1, they are in ampicillin resistant gene. Now, at here, if I cut this from this place, okay, with the BAM H1, which is falls into tetracycline resistant gene, that means this tetracycline gene will become inactive, which is known as insertional deactivation or insertional inactivation. Means if you cut it, if you insert any DNA over here and make rDNA, this tetracycline gene become inactive. That means it won't be remaining active, and the bacteria or whatever it is, but normally we taking this from the bacteria and bacteria have this, so bacteria will lose the tetracycline resistant capacity. That means if you give tetracycline, they will die. But this gene of ampicillin remains as it is intact. So, then for which antibiotic it remains resistant? So, it is inactive, remains for ampicillin. So, ampicillin is your answer. Okay, tetracycline resistant remains as it is. Here, my mistake over there, it's a mistake, not that. Over here, from the diagram, and again this is from the diagram we are doing it. Okay, from the diagram. So this is tetracycline remains inactive, so forget it. Ampicillin remains as it is, remains resistant from ampicillin gene. That's it. Directly. Question 23. The method of producing thousands of plants through the tissue culture is known as micropropagation. This is old syllabus, deleted chapter. Okay, this is deleted chapter. If you remember, you may not have learned this chapter. Chapter 9, strategies of enhancement in the food production. With the page number I have provided. Okay, at the back side, I think. Yes. See, at the back side, this is cutting of the textbook. In that chapter, topic 9.4, tissue culture, where I can see this. Micropropagation. This is your answer. But this is deleted chapter. This is from deleted chapter. So, good luck. Question 24. Choose the correct equation of exponential growth. Again, from the diagram I can say this is J shape, means exponential growth. Answer is here. dN by dt is equal to Rn, which is A. Answer is A. This is sigmoid curve. Okay, this is S shape sigmoid curve, and this is J shape called exponential growth. So, diagram is important. Next question, question number 25. Very good. Question number 25. Arrange the following steps in the process of decomposition. Now, there are many steps and it's a long process. I am not going to read, but see, This is from the textbook directly given. In detrivorous, the breakdown of detritus into smaller particles. The process is called fragmentation. Okay, by the process of leaching. Means what do you see? Fragmentation, leaching, catabolism, then humidification, etc. These are the steps we can find. Now, wherever you see the steps in a way, like see, first fragmentation, fragmentation, fragmentation. Always fragmentation is the first. But in all the four options, fragmentation is given. Now, what will be the next step? Leaching. Now, water soluble organic nutrients go down into soil horizon and get precipitated as unavailable salt. Bacterial and fungal enzyme degrade the detritus into simpler inorganic substances. This process is called catabolism. Now, the second step I can see is leaching, and the third step I can say is catabolism. So second is leaching. Now where do you see leaching is a second? In option C and in option B. Two places. That means you can directly ignore these two. Option B, option D, ignore directly. Okay. Now next, the third is, where do you see catabolism? Here there is no catabolism, but here I can see. That means my answer is A. Fragmentation, then leaching, then catabolism, then humidification, in the end mineralization. So that is your answer. Question 26 in India. How many wildlife sanctuaries are there? Answer is 448. 448 sanctuaries are there. Just a second. Question 26. This is question 27. Okay. Assertion and reason type of question. Assertion A. The microsporangia develop further and becomes pollen sac. Means microsporangia develops further the or turn into pollen sac. Pollen sac. And reason, innermost layer of the microsporangium is tapetum. Now, this is also correct. This is also correct. Means both sentences are correct. But let's see what are the options given. A. A is correct. R is wrong. But R is correct for us. Okay. Because the very inner layer is tapetum. So this is not. B. A and R both are correct. And R is not correct explanations of A. Now this is kind of answer. Let's see other options too. A and R both are correct. Both are correct. But R is the correct explanation. Actually, okay, not correct explanation. And A is wrong, R is correct. But R is, both are correct. So this is not option. This is not your option. Answer is B. A and R both are correct. And R is not correct explanations of A. You can see R does not explain A. So answer is B. Question 28. Government of India legalized MTP in, see, India legalized MTP in 1971. 1971. That's it. Directly. Which process is used when we want to know the genotype of an unknown flower? Now understand the question first. Which process? For you, if you want to know what the genotype, you want to know the genotype, but you know the phenotype of unknown flower. Now here the answer is taste cross. But if you remember, when you know the phenotype, okay, when you know the phenotype and you want to know its genotype, okay, then what we do? We do taste cross. So answer is taste cross. Okay, in here, next question 30. Question 30. Which of the following answer is correct if the template chain of your DNA is having the sequence TGCA of nitrogen base, then what will be the correct sequence of nitrogen base in its mRNA? What I see? 5' to 3' and 3' to 5'. This above strand is in a blue is called template strand and the lowest one is called coding strand. 5 to 3 is coding strand and 3 to 5 is template strand. Now we call this a coding strand because it has the information of making RNA, okay, correct mRNA. That information is in here and that is why I call it coding strand, but we do not use it to make RNA. Okay, why? Because there is a complementary rule. Now, so we will not be using the coding strand. Instead, we use template strand to make its complementary mRNA. So, if I make its mRNA here, now, because of the complementary rule, this mRNA will be exactly like the coding strand. Simply, and that is why, just a second. Now see, template chain sequence is given TGC. Let me write over here TGC ATGC. This is the template strands sequence. Now this sequence, whatever the sequence of its mRNA. So if I form the mRNA of the template strand, what will be? So mRNA, according to the complementary rule, T will give me A, G will give me C, C will give me G, but A will give me T, but this is mRNA, so not T but U. Yes, then T will give me no change, adenine, guanine, then it will be complementary with cytosine and again guanine. So this is my 5' to 3' mRNA. Okay, so this is here AGC ATG. Yes, AC. Sorry, AC. Okay, ACGU. ACGUAGC. So this is my answer. This answer. This is from chapter 5, molecular biology. Okay. See the complementary, and that is why again diagram, and the same diagram. Okay, the same question with the same diagram. Board has asked many times in last year, 3 to 4 year board paper. If you see, you can find the same question there, but the question is changed, but it is from the diagram, from this diagram. That means the board is asking diagram based question, which is very essential and very important for you. So always do read the diagram. Next question here. Question 31. A single step large mutation. It is called as saltation. But where do you see the answer? Answer is here. See. Saltation. Single step large mutation is called saltation. Next question is 32 number of question. Which of the following is an example of cyanobacteria? Now, according to over here, the answer is oscillatoria. Answer is oscillatoria. But spirulina is also an answer. Okay, spirulina is also an answer over here. So A and B both are kind of cyanobacteria. But I think for this question, the oscillatoria will be the correct answer. Now why I am saying so? This is the explanation. You can pause the video and you can read it. Let me just, yes. Okay, you can read it. What is oscillatoria? It is cyanobacterium. Spirulina, cyanobacterium, a commercial source of the protein. Okay, we use this. spirulina. And this azospirillina and azotobacter, which are the options, they are nitrogen fixatum bacteria, but they are not cyanobacteria, but they are heterotrophic bacteria. Okay, so these are not the options, but there is a tie between the oscillatoria and the spirulina. Both are falls under the category of cyanobacterium. Here, spirulina is sometimes classified under cyanobacteria, but may be grouped with microalgae in some older context. In modern biology, both are correct. But I think board will give correct to oscillatoria only. So the answer for you is oscillatoria. Leave it now. Next, question 33. In the process for the biolistics, which metal is used? Now we use for the biolystic gun tungsten and gold. We use method, the biolystic gun, we use only two things, tungsten and the gold. Now just see the option, iron, nickel, tungsten, silver. We use tungsten here. Okay, that's it. Question 34. Which transgenic animal is being used to test the safety of polio vaccine? Monkey, rabbit, mice and the sheep. Here, vaccine safety. Point number 4. Transgenic mice are being developed for the use in testing the safety of the vaccines before they are used on humans. Transgenic mice are being used to test safety for the polio vaccines. So what we use normally? Mice, not sheep, not rabbit, not monkey. Next, 35. If in any pond there were 60 lotuses plant last year and through the reproduction 24 new plants are added, then what will be the birth rate of offspring per lotus per year? This is similar example, okay, like a textbook. See in the textbook what is given. If in a pond there were 20 lotus, what is given? 20 lotus plants last year. And through reproduction, 8 new plants are added, taking the current population to 28. And so we can calculate the birth rate as 8 divided by 20. Means old and the new one will give you the ratio. So that is how, what is given? 60 last year present plus how many added? 24 new plant added. So total is 84 this year. So this year total become 84. But according to this, what we can do is 24 divided by 60. So this will be your answer. Okay. So do your math. Do it. Now next, question 36. What is the percentage of a photosynthetically active radiation called PAR in the plant of the incident solar radiation? You can see answer over here is from energy flow. 12.4. Except for the deep sea hydrothermal ecosystem, sun is the only source of energy for all ecosystem on earth. Now we know this, a sun is only source of energy on the earth. Of incident solar radiation, less than 50% of its is photosynthetically active radiation called PAR. So how much PAR we can see? 50%. If the sun is giving you 100%, for example, a sun will give you 100% of sunlight. Okay, out of that 100% incident sunlight, only 50% is called Okay, PAR means photosynthetically active radiation. And we know that plants and photosynthetic bacteria means autotroph fix sun's radiant energy to make up the food from simple inorganic material, and plant capture only 2 to 10 percentage of this PAR. So out of 50 percentage PAR, a plant will take up only 2 to 10 percentage, means plant can utilize only 2 to 10 percentage of this 50 percentage PAR. So answer is here. The 50 percentage is PAR. Sacred groves found in, again from the textbook, such sacred groves found in Khasi, Jaintia Hills, Meghalaya, Aravalli Hills of Rajasthan, Western Ghat regions of Karnataka and Maharashtra. The Sarguja, Chanda and Bastar areas of Madhya Pradesh. In Meghalaya, the sacred groves are the last refuges for the last number of rare and threatened plants. So the answer from here is Meghalaya. Where you see the Meghalaya here. This is Meghalaya. Okay. So Meghalaya, in the hills of Meghalaya. But see, Assam, Uttar Pradesh and Punjab is not in this answer. So groves are found in Meghalaya. 38 number of question. Which of the following is possess only cross pollination? Means only cross pollination is seen. Self compatibility, self fertilization, bisexual. In all, the self pollination can be seen. So this is not our answer. Okay, what we see, only cross pollination. So self incompatibility means this plant is self incompatible. When the self pollen grain comes and falls, okay, it won't germinate. So it is called self incompatibility. So answer is A. But you can see, okay, cutting of the textbook. The third device to prevent inbreeding is a self incompatibility. Okay, next. 39 number of the question. Which method a sperm is directly injected into the ovum? ICSI. Intracytoplasmic sperm injection. Okay, is another specialized procedure to form an embryo in the laboratory in which a sperm is directly injected into the ovum. Okay. Next question, question 40. The code is nearly universal. It is exception for which of the following Drosophila, bacteria, human and protozoa? Now again cutting off the textbook. See, the code is nearly universal. For example, from bacteria to human, the UUU means triplet of nucleotide would code for phenylalanine PHE. Some exceptions to this rule have been found in mitochondrial codons and in some protozoans. So see the answer, do you see the protozoa here? Because in human and in bacteria, it is same. So Drosophila is also among them. So protozoans is your answer. Now next question is question 41. Which of the following is an example of the fish-like reptiles during evolution that went back from land into water to evolve? Okay, went back from the land into the water means the fish came out of the water, then again go back. So what is that example? Tyrannosaurus, Talicosaurus, Archaeopteryx and ichthyosaurus. Now answer is present, but they all fell into form called deposits slowly. Some of these land reptiles went back into water to evolve into fish-like reptiles. So they are first, these organisms came out of the water, okay, and they evolved as reptiles. But some of those reptiles went back into the water to evolve fish-like reptiles. Probably 200 million year ago. An example is Ichthyosaurus. So Ichthyosaurus is what? Ichthyosaurus is fish-like reptile. So answer is D. Next question, 42. The value of BOD, fill in the blank, in polluted water and fill in the blank in the drinkable water. Now BOD is the measure of organic matter present in the water. The greater the BOD of the wastewater, the more its polluting potential. Means pollution water is having high BOD. So polluted water is having high BOD and the drinking water is having low or lowest. But here answer is given high, low. So answer is here. Next, question 43. Which of the following is known as molecular seizures? Okay, molecular seizures. What is molecular seizures? See, molecular seizures means restriction enzymes. So, restriction enzymes are molecular seizure. 44. From the transgenic animal, which human protein is used for treatment of the emphysema? Transgenic animal is making a protein and we are using that protein to treat the emphysema. Now, human protein alpha-1 antitrypsin used to treat the emphysema. This again, this question is also repeated again actually. If you see past few year paper, you will find this out. So, answer is here directly, alpha-1 antitrypsin. We use it to treat emphysema. Next, 45 number of the question. Correct equation for population growth. Now this equation, again cutting of the textbook. See, n t plus 1 is equal to nt plus b plus I square, in the bracket, minus in a small bracket d plus e. So b plus I all over minus d plus e. B for birth. See, B for birth, I for immigration, and D for death, E for emigration. So equation is here. Now wherever you find this equation, at two places plus is not given, but it is very kind of similar. Okay, so at in one look you will not find it out. So very careful in examination. So answer is here. And t plus 1 is equal to nt plus b plus I minus c plus e. Sorry, d plus e. See your, this is your answer. Now next, here, 46 number of question. The annual net primary productivity, NPP, okay, NPP, of the whole biosphere is approximately how much? Answer is here. Whole biosphere is approximately 170 billion tons. 170 billion tons. Next, again, diagram based question. Diagram based question. Now, diagram. You can see here. Do you see the diagram very carefully? This is in your textbook. So what do you see is, what we have to do is open your textbook and go for the diagram. Okay, so this is thing. Now what is this? It is multicarpellary but Apocarpus gynoecium, not syncarpus. This is syncarpus. This is syncarpus, but this is all females are separate than each other. That's why it's called Apocarpus. Remember this. So this is Apocarpus, multicarpellary Apocarpus gynoecium. Okay, answer is A. Multicarpellary because many females are there and all females are separate from each other. That's why it's called Apocarpus gynoecium. Next, 47. B. 47 number B. Okay, just a second. This is for general student 47A and this is 47B for the blind students. Which of the following is not a water pollinated plant? Answer is A. Lotus. Okay, lotus is not pollinated by the water. Instead, it is through insects. Okay, you can see here. Lotus is insect pollinated, entomophily. It follows entomophily. Next, 48. Which one is correct option for sterilization? Now, this is the first one. So, answer is B. Block the transport of the gamete. Oh, 49 number of questions. Then the first non-cellular forms of life could have originated. See, the first non-cellular form of life could have originated 3 billion years ago. 3 billion years ago. They would have been giant molecules. 50 number of question and the last question. Which of the correct option for the treatment of adenosine deaminase deficiency, ADA, means treatment of ADA, which is correct treatment of ADA? Surgery, gene therapy, immunization or radiotherapy? Now radiotherapy we do for the cancer, immunization also for the cancer, surgery also for cancer, but gene therapy we use to do the ADA. Now cutting is at the back side here. Gene therapy, okay, we do this in gene therapy section only. The whole theory is there, but we don't need to read that how it is happening right now, but it is in the section of gene therapy. So answer will be gene therapy. That's it. Now, so that is it. This is it. The paper is covered. Whole 50 MCQs are over. Please let me know your valuable comments. If you want any specific video on a particular topic of biology from standard 11 and standard 12, so please do comment. I will try to do it. Thank you so much. Thank you so much.